First of all, we performed a cluster analysis in order to look at the structure of the data. As you can see in this TSNE plot, patient 1 and OVCAR3 samples form a separate cluster from FCS7 and patient 8 samples. This is actually consistent with the presence or absence of the R248Q mutation in the p53 gene, with patient 1 and OVCAR3 samples carrying the mutant allele and FCS7 and patient 8 samples harboring the wild type allele. We can further confirm the structure of the data by looking at the clustered heat map. Again, we can see that the samples separate into two discrete groups, with the group on the left hand side including all the samples with the mutant allele, and the group on the right hand side containing all the samples with the wild type allele. And you can see that the genes that distinguish these two groups are strikingly different in terms of their expression. It's also interesting to notice that some of these genes are associated, among others, with the p53 pathway and with apoptosis. So next, we looked at the differential gene expression. We used conditions similar to the ones described in the paper, and we compared samples before and after treatment with a peptide that rescues p53 activity. And we performed this analysis separately for each one of the cell groups in our dataset. So we want to focus here only on the first four volcano plots. So first of all, the samples or the groups with the wild type allele showed no difference in gene expression following exposure to the peptide, as seen here and here. On the other hand, the OVCAR3 and patient 1 samples, which do harbor the mutations, showed an increase in differentially expressed genes here and here. So if we focus on the patient 1 and OVCAR3 comparisons, and we perform an intersection analysis, we can see that in this Venn diagram there is a large number of genes, of differentially expressed genes, that are shared between the two pairwise comparisons. And in fact, the majority of genes contained within the patient 1 comparisons are also contained within the OVCAR3 comparison. If you also look at the scatter plots, we can see that the two comparisons are pretty well correlated with each other. So what we did next was to look at gene set enrichment, so to replicate the gene set enrichment analysis that was done in the paper. For these reasons, we focused on the OVCAR3 sample, and we did both a GO term and a KEGG pathway analysis. So we first of all look at the GO terms analysis. We can see here a summary gene ontology graph, and if we then order samples by score, so first of all starting with the terms that are most upregulated, we can immediately spot terms associated with transcription and RNA metabolism. And on the other hand, if we go and look at the most downregulated terms, we can recognize several metabolic pathways as well as energy generation terms. So moving on to the KEGG pathway analysis, again we start looking first of all at the most upregulated pathways, and we can see here the cell cycle pathway as one of the most upregulated, which is consistent with what is described in the paper. And on the other hand, if we go for the most downregulated one, again we have several terms associated with metabolism, which is consistent with what we saw in the GO term analysis. If we then look more in detail at some of the terms discussed in the paper, so for example apoptosis, we can see that even in our analysis, apoptosis shows up as significantly upregulated following exposure to the peptide. And we can also say the same for the p53 signaling pathway. As you can see here, it is significantly upregulated as indeed is the case in the paper. To conclude, we can look at some of the genes in the p53 signaling pathway as done in the paper and see how they behave in the samples harboring the mutation. So we can start with CDK1NA, which corresponds to the P21 gene, and we can see that upon exposure to the peptide, there is an increase in gene expression in both the patient 1 sample and in the patient 1 samples and in the OVCAR3 samples. We can pick another gene in the list of p53 signaling pathway genes. So I'm going to pick this one, which is PMAIP1, known as NOXA in the paper. And again, we can see the same pattern as before, with samples after exposure to the peptide showing an increase in expression of the gene in both patient 1 and OVCAR3 samples.